He'll be presenting on conventional phase separations, kinetics of colloids in active liquids. Over to you.
Yeah, thanks. First of all, I'd like to thank the organizers for putting up this program, nice program. So today I'll be talking about phase separation in colloidal systems. So the movie that you see here is, let me explain what is this movie about. So what you're seeing here are colloidal particles that are suspended in a bacterial suspension, okay? You don't see the bacteria because they're very small. The particles are larger compared to them. The particles are about 15 micron, and the bacteria is about 2 to 3 micron. You don't see them, but then if I take out the bacteria, there is no dynamics in the system, which means that there is no role of thermal fluctuations. These are athermal systems. The whole reason why these particles are moving is because of the kicks they receive from bacteria. And then what you see is that a homogeneous suspension like this is unstable. The colloidal particles, they have this. There's an effective attraction between them, and then as the time progresses, you see that they start forming these clusters, and there's some sort of a phase separation in the system. And then I'm trying to talk. I'll talk about the system, how is this phase separation different from what we have studied so far in equilibrium systems. Okay, so I'll try to focus on certain aspects of that. Before I proceed, these are the people with whom I've worked on this project. So most of the experiment and the data, this is work of Pragya Kushwaha, who was a PhD student. She's moved to the US recently for a postdoc. And yeah, we collaborated with Shraddha Mishra on the theoretical aspects of this problem. Shraddha Mishra and her group, and also Sanjay Puri at JNU. Yeah, so this is what we know from equilibrium systems. This is an example of phase separation from everyday life, vinegar and oil. They don't mix because they don't like each other, okay? So, we can describe this behavior using a simple diagram like this at very high temperature. If you have a binary system, the mixed state is favored, but if you go to low temperatures, you start seeing these binodal and then spinodal curves. And then if you quench the system suddenly into this spinodal domain, the system is unstable, thermodynamically unstable, and it starts to phase separate via spinodal decomposition. This is something that we all know and we appreciate very well. So this is an example of an Ising model that undergoes spinodal phase separation, okay? So simulations. And this is another example, this has attracted a lot of attention in the recent years. How do these protein condensates form within cells? These protein condensates have very important functions without which the cell cannot function properly. So there has been a lot of focus on this membraneless organelles. Okay, so how do they form? And once again, what kind of one of the pictures that has been painted here is it occurs due to phase separation. Okay, and yeah, these are some of the examples where we see phase separation playing a dominant role. And once again, there's a lot of, we very well know how to describe phase separation in equilibrium systems. I'll not go into the details, but I'll just show you that there's something called model B or Carnot-Lard equation. We can describe them on a microscopic, these are continuum equations. We can describe them, and then there is something called dynamic scaling. Essentially, if you look at the phi is the order parameter, it could be related to the density. If you look at these correlation functions, and then you see that over time, these structures coarsen, and then if you find out a characteristic length scale, one can rescale all these correlation functions, and then they fall on top of each other. So this is the dynamic scaling. This is all seen in equilibrium systems, and then if you look at how these length scales grow as a function of time, you'll see that if you have a conserved order parameter, and then where diffusion plays an important role, then you see that these length scales grow with an exponent of one third. Okay, t goes as t to the power 1/3. So that's what we understand. And we also know that these interfaces are very sharp. So if you go look at these correlation functions at very small distances, you start seeing these singularities. And then if you take a Fourier transform of this, you can also see these singularities appearing in these structure factor, which is called, so this is called Perot law. So these are some things that we know, we understand about equilibrium systems. Now we want to see what happens in these systems, okay, in active systems. So this is a simple picture of experiment. We have this bacteria and then colloids mixed together, yeah, so very simple experiment. The first thing that I want to show is the distribution of local densities. What we do is you have field of view, you cross-grain the density and then look at the distribution of the density. At short times, you have a unimodal curve, but then as the time progresses, you see this bimodality coming in, which is a bona fide signature of phase separation in our system. So now we also look at define order parameter, if the density is greater than the average density, then the order parameter is +1, otherwise it is -1. We look at the correlation functions. And very interestingly, the color indicates here correlation function with increasing time. The red is large time since the start of the experiment, and blue is very small time since the start of experiment. As you see, as time progresses, these clusters grow in time, and then once again we can find out a characteristic length scale and then rescale all these correlations. Okay. And if you look at the length scale, how it grows, you see that there is a strong deviation from Lifshitz-Slizov law that we saw in equilibrium systems. So once again, if you have in most equilibrium systems with conserved order parameter, you see that the z 1 over z is one third. But in this case, we find some unconventional exponents, which is 1/4. And this behavior is robust. We have changed the density of the colloids, and we also changed the density of the bacteria, and then it seems to be very, very robust. Okay, so this exponent 1/4, it remains 1/4 over a decade of bacterial densities. And we look at the structure. So that was about how the domain sizes grow in time. That is about the dynamics. And now, if you look at the structure of these domains that are formed, we see that these are very rough domains. These are not domains with sharp interfaces. So to characterize this, what we do is we look at the scale correlation function. It can be written in this fashion. Once again, if you are in equilibrium, most equilibrium systems show alpha close to 1, but here we see that this exponent, the cusp exponent, it turns out to be 0.47. Okay. And one can also look at the structure factor, and instead of porode behavior, we see the strong signatures of non-porode tails. Okay, so here d plus alpha, if you have porode behavior, you will see d plus alpha is -3. This exponent happens to be -3. S of k scales as k to the power -3, and here we see an exponent that is deviating from that porode thing. And using these relations, all these relations have been, how does one relate alpha to the fractal dimension? These are fractal structures, so how does one relate alpha to these fractal dimensions? There are these relations that have been explored previously by these people. We look for, once again, from these exponents, we can figure out that the fractal dimension is 1.5. So what we did was we used box counting algorithm to explicitly calculate the fractal dimension of our clusters, and then indeed we see that the fractal dimension is very close to what we find from these relations and from the box counting algorithm. So indeed, what we are seeing is these are fractal structures. These are not sharp interfaces. So let me come to the coarse-grained model. We can understand all these exponents, the non-trivial exponents that we have seen in experiments using a coarse-grained model. This is model B plus colored noise. This is something that was proposed by these set of people. Once again, let me go quickly. You see this dynamic scaling. The length scales grow as t to the power 1/4. And then you see a cusp exponent, and the exponent is 0.66. And then also, if you look at, you start seeing non-porode tails with similar exponents. Okay. And why is it interesting is because in the recent years, there has been a lot of interest in the community to understand phase transition in purely active systems. There also, they see that these length scales grow with non-trivial exponents, And so these are active model B. They call these models as active model B. And also, there's a lot of discussion about how these exponents deviate from equilibrium systems in the context of FDP, fluctuation-dominated phase ordering. So with that, I would like to, these are the conclusions of my talk. Colloidal particles form dynamic clusters due to interplay of effective attraction and activity. Novel features of active phase separation, slower coarsening of kinetics compared to Lifshitz-Lizov growth in equilibrium. And we find fractal interfaces with cusp exponent. And we've been able to explain all these exponents using, many of these exponents using coarse-grained model where it's a model B plus noise, colored noise. Yeah, with that, I'd like to take questions. Yeah.
And what is this algorithm? You just subsystem number fluctuation.
You divide the system into small boxes of length scale L, and then you find out how many boxes contain the interface, and then you change the box length, and then you contain. Yeah, contain the interface. You count the number of boxes that contain the interface. Yeah.
I have one. So model B supposedly follows Forward's law, no? I mean, K minus 2.
Yeah, model B. So this is a model B plus noise.
Oh, plus
with colored noise. So I mean, what we are saying is that the effect of, I mean, we are mapping this onto a two-component system wherein the bacteria, like, acts like noise, and it's a colored noise essentially. It's not thermal noise. Yeah, it's colored noise.
Do you have an estimate of the kind of interactions that that is being mediated by the bacteria? I mean, it should be short range, right? The growth law that you're showing, it's about the short range. I think in your class, the concept dynamics growth law that you were showing.
Yeah.
Those are for short range. I think in university class, right? Short range.
So that I still have to. I mean, I don't have a good answer for that. Yeah, maybe we can discuss this later. Yeah. So I mean, so here in this system, like the colloidal particles are very much larger, are much larger compared to the bacteria. So their time scales of motion are there's a time scale separation. So we can treat the motion of the bacteria as some sort of a colored noise.
That can be directly also just measured?
Probably, like, one can do some optical tweezer experiments and try to characterize the noise that you see in these systems. And
just from images, just from images.
From the images, the noise, probably we need to image the bacteria then, like, if you want to characterize the noise that they are, yeah.
So how did you generate a colored noise in your simulation?
Okay, that's a good question. Pratiksha is here. Yeah, you can ask her. She'll tell you all the details because she's the one, like, who has done the calculations. Yeah.
I have a quick question. I want to know, this is for what concentration of colloidal particle?
So it's about 0.2, let's say, and we have changed the density of the colloids. This is 0.23, 0.3. Like, it's still not very dense. So you see this phase separation over a small range of area fractions, because if you go to very large area fractions, then you start seeing percolation and all. You don't. You see it's a different phenomena. Yeah. Yeah. There's a range of densities where you can see this phenomena. Yeah. 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 The clusters break and form, yes. Okay, yeah.
That cusp exponent is related to the cusp exponent that you find for the spatial decay.
Okay, I can talk to you later.
Experiment, is it possible to measure the autocorrelation?
Yeah. I mean, if you are just talking about density autocorrelation, we can do that. Yeah.
It might be.
Yeah.
So let's thank speaker.